Okay, well, thank you for the introduction and allowing me to talk. I'll try to keep this brief because my time would now run into lunch. Many of the same figures on here are on the now award-winning poster that is down the hall. I'm happy to talk over lunch and we can go through some of those. So basically, piggybacking on Peter's general introduction to sequencing normal tissue, we'll say all the same stuff applies in breast. Obviously, there are different risk factors in breast as compared to lung. So kind of one of the goals of what we want to do is similarly look at how the normal breast tissue is different across the relevant risk factors in breast, namely age, germline BRCA status. So there's about a 12% lifetime risk of developing breast cancer generally for women. If you're a BRCA1 or BRCA2 germline carrier, it's up to about 80%. And then there's an interesting relationship with pregnancy, though I'm not going to talk about that in this presentation. And then drivers, signatures, clonal structure, et cetera. So the study design we're doing is we're getting normal breast tissue from kind of 3 different groups: reduction mammoplasty, so as close to normal as we can get. That's on the far right side. So we have the youngest patient we have is 19 and the oldest is 68. Prophylactic mastectomy from BRCA1 and BRCA2 carriers. These tend to be skew younger. I thought there were points on that graph at some point, but I guess not. And then mastectomy samples from cancer patients. So we're getting both tumor and normal. So kind of the goal is to get about 20 whole genome samples per patient, 10 samples per category. We're a little over halfway there at the moment. I'll be showing about 100 samples from this. So we're using a technique called laser capture microscopy, which was mentioned yesterday and just a second ago by Peter, using a laser on tissue to cut out a thing. In case I was wondering, this is what it looks like if you do the thing and this doesn't freeze, which we will see. Come on, maybe, no. Okay, anyway, the pathologist will recognize that this is not breast tissue. This is actually lung tissue. Oh, hey, there we go. And so you go and you highlight a little region of interest, and you then click on the machine, and then a little laser cuts it out and drops it down into the wall behind. This is really exciting the first 1,000 times you do it. After that, it becomes less so. This is about 10,000 square microns, 160 or so cells, maybe about 1 nanogram of total input material. We can reasonably reliably turn this into a whole genome. So we get breast sample, snap frozen biopsy. We use PaxGene, which is an alcohol-based fixative, embed in paraffin, section, and H&E stain. So it gives us something that is about as good as formalin-fixed paraffin-embedded material without all of the problems of formalin affecting the DNA. Then go through the LCM process, take pretty pictures of it. So to date, I've done 1,200 wells for breast, and a whole bunch more for a lung project. Those go into a low-input DNA prep pipeline that importantly doesn't have any whole genome amplification. And then in breast, we do a 2-step approach, initially a targeted screen to look for interesting driver mutations at higher depth and assess the clonality of the sample. And then those that are deemed sufficiently clonal and or sufficiently interesting go on to whole genome sequencing. So there's 700 that have been targeted to date, 355 are somewhere in the whole genome queue. I'm showing you about 110 or so. So I mentioned clonality. Why is that important? So this is a sample actually similar to the bronchial epithelium I showed the LCM photo of, where we have a nice clonal VAF. So median's around 0.5. You have 2 copies of every gene. One of them's mutated. Half of the alleles are variant. So the variallele frequency is 0.5. Why is that nice? The blue are the real mutations. The red are the suspected ones. So there's a nice distance between the two. We tend not to see that in breast. So for the most part there, the mean VAF is around 0.25, so likely a couple different clones are giving rise to the sample. That means that the distribution between the real mutations and the potentially artifactual ones are a little closer. It has a bit more caveats with the data, although we do occasionally see samples where there's kind of a large clone around 0.5 and some subclonal ones lower. So it's a mixed bag, and this is why we do the 2-step approach. If we just went straight forward with everything, we would end up spending a lot of time sequencing things that can't be sequenced. So the distribution of VAFs across the cohort, you could see that there's considerable heterogeneity, so each color is a different patient, where Pretty much all of the patients have some samples that are in the 0.3 to 0.4 range as their kind of upper mean VAF, but there are also examples from all of them that are on the lower side, to the point where we have, down here, the majority of the sample is like 0.15. This is an example of the first approach where we just sequenced everything, and this is why we do a two-step. Whereas this one is, nice and clonal, and you can imagine that we're able to much more easily and be confident in the mutations we call here as compared to here. And importantly, there doesn't seem to be a huge difference in clonality between the reduction mammoplasty samples or the BRCA carriers over here. So a couple of obvious, this is not going to like my colors, is that down here we have normal breast tissue, so the LCM stuff. Up here are some breast whole genomes. This is the mutation burden in the normal breast tissue. It's about 0.3 per megabase, so about 1,000 mutations in a genome, as compared to what a typical breast tumor is, about 3,000 mutations. And you can see that in the median, maybe you can't see, but in theory you could, that there's a line here that kind of overlaps with the lower. So there's normal breast samples that kind of overlap with the lower end of the distribution of tumors, and some of the highest mutation burden in the normal breast overlaps with kind of the median of breast tumors. Kind of the big exciting first takeaway is that if you compare the BRCA carriers to those that are wild type, there is no difference whatsoever in their mutation burden. If anything, it's slightly older in the wild type, but this is explainable by the fact that cohort's a tad older. And then there's this group in the middle of a couple of cancer patients that I'll talk about more in detail later with a very high mutation burden. So this argues pretty strongly that there doesn't seem to be an impact of haploinsufficiency for the BRCA mutation. If we regress this with age, you can clearly see that in red we have BRCA carriers, and in yellow are the breast reduction normal ones. They don't seem to, hop on either side, so they're nicely intermixed. We have a couple patients, two patients that are the same age that are nicely within each other, and this patient here has cancer, and so even with BRCA and being someone who develops cancer, that doesn't deviate really at all from what the line that you would expect. So the overall mutation burden is about maybe 15 per year, which is about half of what you saw Peter present just a second ago. Interestingly, including in endometrium, which is another female estrogen-responsive tissue, but you could also imagine that endometrium cycles much more quickly and frankly does a lot more than breast does for most of its life. So a couple other quick takeaways. Driver events are infrequent, so this is looking at copy number. And so most of all the samples, basically all of the ones of BRCA carriers and most across the other samples are copy number normal. There's no copy number changes whatsoever. You do get occasional single events, and 1Q gain is the most frequent. This is the most complicated copy number situation in any of the samples we have, with 1Q gain and 16Q loss, both things seen in breast cancer. And this actually happens to come from one of the reduction mammoplasty samples, so an otherwise normal individual. That doesn't work. Okay, well, if there are a bunch of points on here, and there's a bunch of points on the poster, that it would show you that there's not a whole lot of mutations in BRCA1 or BRCA2 that would be likely second hits. There are a whole bunch of mutations in PIK3CA, some in PI3R1, so both PI3 kinase pathway mutations, and some P53 mutations as well. So GATA3, PIK3CA, PIK3R1, and P53 are all under significant selection via Inigo's DNDS algorithm, and there's evidence that PIK3CA can lead to clonal expansion, and either through copy number or point mutations, it doesn't look like there's any second hits to BRCA. Doing mutational signatures on this set of samples, we end up pulling 8 using SIG Profiler. The first 2, 1 and 5, we see in basically all normal tissues. We do pull out 2 and 13, so Applebeck. Signature 18 is reactive oxygen species. Some of those are real. Some of those are going to be artifactual due to the extra PCR cycles we do in our pipeline. And then this really interesting one is signature 25, which has only been seen previously in Hodgkin lymphoma cell lines, and I'll talk about that in a second. And then weirdly, a UV signature that I think only occurs in samples with this, so these two are probably related, and that's just noise. But noticeably absent, there's no signature 3, which is the one that is in BRCA patients, in BRCA cancers, rather. So if we now look at the activities across the cohort, what jumps out immediately is there's lots of red and lots of gold-ish. So signatures 1 and 5 are predominant across all of them. If you compare the BRCA to the breast reduction samples, the ratios of 1 and 5 look identical. The burdens between the two look more or less identical. There's occasionally a little bit of signature 18 that pops on some of those, and the weird 52 that looks artifactual. And then if you look in the middle here, in the cancer patient, there's this whole bunch of pink, which is all attributable to signature 25. And you can see that I didn't include it in the age regression because this is such a clear outlier. Whereas the burden here is around 1,000 mutations. Here we get 2,000 to 3,000 mutations in a whole genome, almost all of that driven by the signature 25. So much like what Peter just showed, we can take all these samples, put them in a tree, overlay the mutational signatures. And so this is that cancer patient that was, turns out at age 17, they had a lymphoma. That's all the annotation is. I'm going to assume it was a Hodgkin's lymphoma. I'm going to assume they got treated somehow, and they responded positively to it. And then 25 years later, they got breast cancer. And so in this pink color is the signature 25, and you can see that in both so these are 3 samples taken from the tumor, and then in addition to a whole series of samples taken from the breast, huge increase of mutations occurred due to the signature, and then kind of both in the main part of this branch, and then after in the splitting, you have quite a bit of APOBEC activity in the tumor. APOBEC is more or less absent from the rest of the samples. So kind of what's all going on? Up here, we have 2 hits to P53 that happen along this long branch of the tumor, both of which, certainly the top one, possibly the bottom one, are attributed to signature 25. So if 25 is something to do with the actual treatment the patient got, which would make sense as it's affecting kind of all of the tissue in the patient's body, at age 17, they get blasted with some combination of chemo, possibly radiation as well. That's inducing a whole series of mutations, including 2 mutations that look like they go on to help contribute to this tumor. Also interesting is that if we now look at the actual tissue, so here we have a nice pretty H&E stain, the little circles are what was actually cut out. So these are about 40 to 80 microns in diameter, so an individual acinus from a breast terminal lobule unit. Nicely, the samples taken from the same structure all contribute to the same structure in the tree. That makes sense. But here we have something kind of interesting where this red group and this yellow group are only about 500 microns apart. But you see they deviate quite early on in life, and there's a whole series of these signature 25 mutations that pop up in each branch. So that means, this whole structure was laid out before the age of 17, or at whatever age they got, whatever treatment they got in response to the chemotherapy. And then one final patient, before I wrap up. This is a BRCA carrier that has ER-positive breast cancer. They've had it 3 times in their life. It was initially missed via BRCA screening and found at time number 3. They were previously treated with an aromatase inhibitor. If we look at the drivers in this patient, on the bottom we have the tumor, much higher burden. We have PIK3CA and the aromatase inhibition resistance mutation, ESR1, both of which likely caused by signature 25. And then in a large group of the patient's normal breast, up here, we have the exact same PIK3CA mutation. This is the only mutation shared between the normal tissue and the tumor. And these are quite a distance away from each other. I have no idea how far away from each other they are, but they aren't on the same tissue block. So at the very least, centimeters. Which means it's probably quite unlikely that this is the actual same mutation. I mean, it's the same physical location in the genome, but it probably happened independently, as this is the most common mutation in PIK3CA in breast cancer, and it's also driven in both cases by the most active signature in both of these samples. And so interestingly, there's about a 3 millimeter diameter region of the normal breast that all has this same PIK3CA mutation. These samples tend to be more clonal than comparable ones. And in this is an area here which is slightly metaplastic, and then an area over here, which has been targeted sequenced, not whole genome, which is why it's not on the tree, that looks completely normal. So we have maybe some evidence of, this leading to expansion and maybe possibly driving a little bit of change over there, but more to investigate moving forward. So overall conclusions, 15 mutations per year, mostly signature 5. No difference between BRCA carriers and those that are wild type. PIK3CA's doing a bunch of stuff in the normal breast, along with PTEN, PIK3R1. And then we can see the previous lymphoma had some sort of impact in the normal breast tissue, so that's going to motivate future studies looking at pre-treatment in both breast and a whole bunch of other samples where we can learn things about how the tissue developed. So with that, lots of people to acknowledge, Sanger, others. Thank you, San Diego, both for the algorithms and hosting us in the winter so I didn't have to be in Cambridge. So thank you.